Alright, you guys. So yesterday we began with our genetics unit and we discussed things like Mendel and his experimentation with pea plants. So today we're going to apply what we learned yesterday to a thing in genetics known as a Punnett square. Now a Punnett square is going to predict offspring ratios such as genotypic and phenotypic ratios. Alright, now yesterday we talked about some things known as alleles. And there are two different types of alleles that we're going to discuss using Punnett squares. And those are going to be dominant alleles versus recessive alleles, okay? Now when something is dominant and it is present, it's always going to be favored, okay? And whenever you have a dominant allele, it is also always represented by a capital letter. And we'll discuss what these letters mean in a few minutes. Now capital letters can be any letter you want, such as big B, big C, big G, or big A, okay? Now, if it's not dominant, it's going to be called a recessive allele. And recessive alleles aren't favored in a Punnett square or in a cross unless they're homozygous. And I know you don't know what homozygous means yet, but we'll discuss that in just 1 second, okay? And a recessive allele is always represented by a lowercase letter, such as big B, little c, little a, little d, okay? Alright, now I know you're wondering what exactly these letters I'm talking about mean. These letters are going to represent whether an allele is homozygous or heterozygous. If it's homozygous, it means that both alleles are the same. Remember that homo means same. And examples of those would be big B, or little b. Notice how each letter in the pair is either both uppercase or lowercase, okay? Now, it can also be heterozygous, which means both alleles are different. So that would be like big B, little b. Alright? Now, I'm going to show you how you would apply these. Whenever you're making a Punnett square, there are a few rules to follow. The first rule is always going to be to identify your genotypes. Your second rule is going to be to set up a table for a cross. Your third rule is always going to be to label each part, or the top and the bottom, or the left and the right, or the top and the left, with a pair. And step 4 is always to predict your genotypic and phenotypic ratios. Now I know you're wondering what in the world I'm talking about, but the only true way to get good at Punnett squares is to do them. So we're going to do a couple of examples. Alright, so whenever you're doing Punnett square problems in genetics, they're usually always going to be a word problem. And they're usually always going to have like a paragraph format. And your job is to always pick the words that you need, or the words that are important, or the keywords. Alright, so let's do this one together. An individual that is heterozygous for a particular gene is crossing with an individual that is also heterozygous for that gene. Complete the Punnett square and predict the phenotypic and genotypic ratios. The particular rate is dominant, okay? Now I've already underlined the important words that you would need in this example. You know that an individual is heterozygous, another individual is heterozygous, and they're looking for a Punnett square and for you to complete the phenotypic and genotypic ratios. And it also tells you that the trait is dominant, okay? So let's see what this would look like if we showed our work. Since we know that both individuals are heterozygous, we're just going to assign them big B, little b. It doesn't matter what letters you assign alleles, just make sure that the capital and lowercase letters usually don't look the same. So like with big C, little c, those kind of look the same, so don't get confused. Use letters that look different. Alright, so we know both individuals are heterozygous, which means that each allele is going to be represented by a different letter. So that's why we have big B, little b, and big B, little b. Alright? Now, for steps 2 and 3, I told you to set up a table and label each side of the table. So, we have a big, we're going to put, just like I have here, just do a square, that's where the Punnett square comes from. You're going to have a big B, little b on the top, and a big B, little b on the left, okay? Each pair gets a different part of the square. So like big B, little b would go right here, and this big B, little b would go on the side. It doesn't really matter which order and which side you put these, okay? Now, what you're going to do is just like multiplication almost. You're just going to bring each letter down and across, okay? So like with big B, you just bring them down together. with big B, little b, they're going to come across together. Same way right here, big B, little b together, and little b together, okay? Now that we have our Punnett square complete and filled in, our fourth step is going to be to make our ratio, okay? Now, remember I told you phenotypic ratio needs to be thought of as a picture or how something would look. We didn't discuss in the example exactly what gene we were talking about. We just know that the trait is dominant, okay? So, you can see here I told you capital letters always represent dominance, okay? So in this case we have 1, 2, 3 sets of alleles that have a big B. So that's going to mean we have 3 dominant, okay? And then we have one that's little b, so that's going to be a recessive, okay? And the way to always figure these out is that you can add, so 3 plus 1 is 4, and we also have 4 squares, okay? Now for the genotypic ratio is always going to be the alleles with the letters. So you just look in your Punnett square for these. So we have 1 big B, so 1 big B. We have 2 big B, little b's, so that's 2 big B, little b's. And we have 1 little b, so that's 1 little b, okay? And these are the same way you can add these up. 1 plus 2 is 3, plus 1 is 4, and we have 4. Now sometimes when you do these, they're going to ask for percentages, okay? But we'll talk about that tomorrow in class. Alright, so now I want you to try. An individual is homozygous for a gene and is crossing with a heterozygous individual. The particular trait is dominant. Complete the Punnett square and predict the genotypic and phenotypic ratios. Now remember your rules when you're doing this example, okay? Remember to do 1 through 4 and you should be fine. And make sure you actually do this because tomorrow in class I will be checking these along with your notes. And tomorrow in class we're also going to be doing tons of examples so you can get more practice and get better at these, okay?